Have you ever wished you could just delete a typo from your body's instruction manual? A tiny mistake in your genetic code that leads to a serious disease? What if I told you we were on the verge of having a tool that can do exactly that? A tool so powerful it could not only cure inherited illnesses, but change the very future of our species. This isn't science fiction. This is CRISPR. So, what exactly is this thing with the catchy name? CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is essentially a pair of genetic scissors. Imagine your DNA as a massive book, the encyclopedia of you. This book contains all the instructions for building and running your body. Sometimes, there are spelling errors in this book, tiny mutations that can cause diseases like sickle cell anemia, cystic fibrosis, or Huntington's disease. For the longest time, we could only read this book. We could identify the typos, but we couldn't fix them. We were stuck with the text we were given. Then came CRISPR. Originally discovered as a quirky defense mechanism in bacteria, scientists figured out how to harness it. Bacteria use it to chop up the DNA of invading viruses. Like a microscopic immune system, we've reprogrammed this system to find a specific sequence of DNA in any organism, including humans, and make a precise cut. Think of it like the find and replace function in a word processor. The CRISPR system has two key parts. The first is a guide molecule, like a search query, that we can design to find a very specific sentence in our DNA. The second part is an enzyme, most famously Cas9, which is the pair of scissors that does the cutting. Once the guide finds its target, the Cas9 enzyme snips the DNA at that exact spot. And this is where the magic happens. When our cells notice a break in their DNA, they rush to repair it. We can take advantage of this natural repair process. We can either let the cell patch up the break, which often disables the gene, useful for turning off a harmful gene, or, and this is the really exciting part, we can provide a new correct piece of DNA. The cell's repair machinery can then use this template to replace the old faulty sequence with the new, healthy one. It's like highlighting a typo, deleting it, and typing in the correct word. The potential applications are mind-boggling. We're already seeing incredible progress in medicine. For diseases caused by a single faulty gene, CRISPR offers a direct cure, not just a treatment. Take sickle cell anemia, a painful and life-shortening blood disorder caused by one tiny mutation. Clinical trials are already underway, where doctors take a patient's own blood stem cells, use CRISPR to correct the mutation in a lab, and then infuse these edited, healthy cells back into the patient. The results have been life-changing, with some patients now living since. This could be the future for thousands of genetic disorders. We're talking about muscular dystrophy, Tay-Sachs, and certain types of inherited blindness. In one experiment, scientists even used CRISPR to successfully remove HIV DNA from the genomes of living mice. The possibilities for treating cancers, which are driven by genetic mutations, are also enormous. We could edit a patient's own immune cells to make them superpowered cancer hunters. Beyond medicine, CRISPR is revolutionizing agriculture. We can create crops that are more nutritious, resistant to drought and pests, and have a longer shelf life. This could be a game changer for global food security. Imagine wheat that doesn't contain gluten for people with celiac disease, or peanuts that don't cause allergic reactions. Scientists are even exploring using CRISPR to bring back extinct species, like the woolly mammoth, by editing the DNA of its closest living relative, the Asian elephant. It's a project called de-extinction. And while it's incredibly complex, CRISPR makes it theoretically possible. But with great power comes great responsibility, and this is where the conversation gets complicated, and frankly, a little scary. The real ethical minefield isn't just about curing diseases in a living person. It's about something called germline editing. So far, we've been talking about somatic editing, which means editing the cells of a specific part of the body, like blood or lung cells. These changes affect only the individual patient and are not passed on to their children. Germline editing, on the other hand, involves making changes to sperm, eggs, or embryos. These changes would be permanent and inheritable, passed down through all future generations. This is where we cross a major line. On one hand, germline editing could eradicate a hereditary disease from a family line forever. A family cursed with Huntington's disease could ensure their children, grandchildren, and all their descendants would never have to fear it. That sounds amazing, right? A gift to future generations. But the flip side is daunting. What if we make a mistake? An unintended edit could create a new, unforeseen health problem that would then be permanently etched into the human gene pool. We simply don't know the long-term consequences. Our DNA is incredibly complex, and genes often have multiple functions. Fixing one problem might accidentally create another one, decades down the line. And then there's the even more controversial topic, enhancement. If we can remove genes for disease, why not add genes for desirable traits? What about genes for higher intelligence, greater athletic ability, or a specific eye color? This leads us down the path to designer babies. This raises profound questions about equality and what it means to be human. If gene editing for enhancement becomes available, it will almost certainly be expensive. This could create a genetic divide, a new kind of class system where the wealthy can afford to give their children genetic advantages, while others cannot. We could end up with two distinct human subspecies, the genetically enhanced and the naturals. It's a dystopian scenario, but one we have to consider. Where do we draw the line between therapy and enhancement? Is preventing a predisposition to Alzheimer's therapy? What about preventing nearsightedness? What about making someone taller? The lines are incredibly blurry. In 2018, the world was shocked when a scientist in China announced he had created the first gene-edited babies, twin girls, with the goal of making them resistant to HIV. The scientific community reacted with widespread condemnation. The experiment was seen as unethical, irresponsible, and premature. It violated international consensus that germline editing in humans should not be attempted at this stage. It was a wake-up call that the technology is moving faster than our ability to grapple with its ethical implications. So, where do we go from here? CRISPR is a tool, and like any powerful tool, it can be used for immense good or for purposes we might deeply regret. The scientific debate is no longer just about can we do this, but should we do this? It requires a global conversation, not just among scientists, but involving ethicists, policymakers, and all of us. We need to establish clear, international guidelines and regulations. We stand at a crossroads in human history. CRISPR gives us the potential to take control of our own evolution, to rewrite the very code of life. We could be the generation that ends genetic disease forever, but we could also be the generation that opens a Pandora's box of unforeseen consequences and deepens social inequality. The power is in our hands. The question is, how will we choose to use it? Thanks for watching. This is a massive topic, and I'd love to hear your thoughts in the comments. Where do you think we should draw the line? If you found this video thought-provoking, please hit that like button and subscribe for more deep dives into the science that is shaping our future. See you next time.